Okay, after completion of the first bit, we are start to the short answer questions. So these are the complete 4 marks questions. So these complete 4 marks questions are 8 questions. In these 8 questions, you should write 6 questions. Remaining 2 are the choice. Right. So in our first model paper, the 11th question is right. For example, if you take a butter, how does and absorb into the intestine? So we are explaining about that first of all. Right. In our food containing number of fatty acids and fats, especially fats, lipids. So these fats are how these are digested in the small intestine. So the pattern especially. So majorly here the digest of fats, especially butter, like these lipids, these are digested by the bile juice actually. So this bile juice is secreted by the liver. So this bile juice does not have any digestive enzyme, but it is involved in the digestion of fat. So we are discussing about the how the bile juice is involved in the digestion of fats, right? So the bile juice containing to the especially these are bile salts. These bile salts. So this example for the bile salts, these are the glycocolloids and tarocolloids. So these glycocolloids and tarocolloids having a sodium and potassium molecules especially. So these glycocolloids and tarocolloids are also called these bile salts. These bile salts are used for the emulsification of fats. So what is the meaning of emulsification? Emulsification means here it is the complete complex fat molecules are converted into the these are the means here large sized fat molecules are absorbed, these are converted into the small fat molecules. It is the process of emulsification. So after emulsification, these small molecules of these these small molecules are under reaction with the lipase enzyme. So this lipase enzyme also secreted from the intestinal cells. That's why lipase enzyme is called intestinal lipase. And finally, these after completion of the emulsification, these emulsified fats are undergoes to the intestinal lipase, and these these fats are converted into the emulsified fats. So these emulsified fats are undergoes to the lipase. After that, these emulsified fats are converted into the triglycerides, diglycerides and monoglycerides. Finally, these di, and monoglycerides are also undergoes to the lipase enzyme. Afterwards, these are finally absorbed, these are finally converted into the glycerides, and these are especially fatty acids. So it is the process of digestion of fats. So first of all here the emulsification. Emulsification means firstly here these are the fats. So these fats are undergoes to the bile salts. So example for the bile salts, these are the glycocolloids and tarocolloids. So these glycocolloids and tarocolloids having a sodium and potassium molecules especially. These sodium and potassium molecules are emulsified to the fats into the emulsified fats, right? Afterwards, these emulsified fats are undergoes to the lipase enzyme. So actually this lipase enzyme is secreted in the intestinal cells. That's why this lipase is called intestinal lipase. Afterwards, these emulsified fats are converted into the these are triglycerides and diglycerides and these are the monoglycerides and finally some are also converted into the fatty acids. It is the main thing, first thing. So afterwards, if the monosaccharides are present, these are the another emulsified fats molecule after completion of the reaction. First that is intestinal lipase. For example, these monoglycerides. These are also undergoes to the intestinal lipase, and these are final products. These are forms to the final products. These are the glycerols and fatty acids. It is the process of digestion of fatty acids. And finally, these glycerols and fatty acids, how does absorbed into the intestinal villi especially? Generally, these glycerols and fatty acids does not enter into the capillaries. Why? Because these glycerols and fatty acids has a long peptide chains actually. So these are converted into the firstly micellase in the mucosa of intestine. So these micellase are absorbed into the mucosa of intestine, right? So by the lacteals especially. So these lacteals are the one of the lymphatic vessels. These lacteals are one of the lymphatic vessels, right? And finally, these glycerols and fatty acids are firstly enter into the intestinal mucosa, and in the intestinal mucosa, these glycerols and fatty acids are converted into the micellase. These micellase are all absorbed into the lymphatic vessels by the lacteals. Finally, these lacteals are open into the large lymphatic duct. This large lymphatic duct is open into this subclavicle vein, that is the clavicle vein. It is open into the clavicle vein. Finally, these micellase are enter into the bloodstream at the left subclavicular vein. So it is the main thing. During the circulation, where these micellase are present in the circulation, these are converted into the chylomicrons under the lipoproteins especially. So finally, these fats means these simple diffusible molecules are the glycerols and fatty acids are enter into the bloodstream as a micellase and also the chylomicrons. It is the main thing of digestion of fats. It is the main thing. So once again I will repeat that. So complete fats are emulsified by the bile salts. These bile salts are present in the bile juice. So the best example for the bile salts are glycocolloids and tarocolloids. So the glycocolloids and tarocolloids has a sodium and potassium molecules. So the sodium and potassium molecules are emulsified to the fatty acids especially. So these fats are bile salts undergoes to the emulsification process. Finally, emulsified fats are formed. After that, these emulsified fats are undergoes to the intestinal lipase enzyme. Finally, these are converted into the triglycerides, diglycerides, monoglycerides, and end products. They are the fatty acids and also glycerols. For example, after these monoglycerides, final products, these monoglycerides are formed to the final end products. So these monoglycerides, triglycerides, and diglycerides, these are also reacted by the lipase enzyme. Finally, these are formed to the simple diffusible molecules. These are the glycerol and fatty acids. So basically, these glycerols and fatty acids does not enter into the bloodstream directly. So these are firstly enter into the intestinal mucosa and these are converted into the micellase. So afterwards, these micellase are enter into the bloodstream through the lymphatic vessels by the lacteals especially. During the circulation, these macellus are converted into the chylomicrons. Finally, these are entered into the bloodstream at the left subclavicular vein. So, it is the process. Another one is, that is a 12th question, right? Example, that is explain the contractile protein. So it is from the muscle contraction, skeleton and muscle contraction, right? So contractile proteins. What is the contractile proteins which are involved in the contraction of muscle especially? So firstly, the example for the contractile proteins are, that is, first one is actin, second one is myosin. These are called contractile proteins. These actin and myosin, these are present in the sarcomere. Sarcomere. So firstly, what is sarcomere? Sarcomere means these sarcomere present between two jet lines or two Dobby lines in the muscle fiber. So it is called sarcomere. So two Dobby two jet lines. Two jet lines having a which type of structures? So these structures are called, one is actin, second one is myosin. These are also called light bands and thick bands. These are also called I band and an isotropic band, A band. Right here, I band is nothing but actin, A band is nothing but myosin. So mainly these contractile proteins have a main role in the formation of muscle contraction and relaxation especially. Right, firstly we are discussing each and every one. Firstly, that is actin. So firstly we are draw a small diagram, that is sarcomere. Right here these are the sarcomere structure, that is a muscle fiber. So it is the actin, means that is a thin band. So these are the Z lines, right? And here middle part of this sarcomere, these are the myosin bands. These are also thick bands. These are also called thick bands. Dark and light bands are also called this. Right. Between these center line is called M line. M line means it is a middle line. And this space is called H zone. H means that is Hansen's zone, right? And here, so these parts are the isotropic, means these are also nothing but thin bands, also nothing but actin bands. And these are that is myosin. These are the myosin band. And between this place, from this place to this place, is called A band. A band means anisotropic. This A band cannot allow to any light from this region. And isotropic means this region, from this region to this region, these are thin bands are allowing to the light. That's why these bands are called I bands, means isotropic bands. So it is the sarcomere present between two Z lines. It is called sarcomere. So here the contraction and relaxation of these contractile proteins, the muscle shows to that is a rigid and a relaxation region. So this I band it is also called actin, and it is called myosin, it is the dark band, right. So these are the contractile proteins. Actually the structural functional unit of muscle is sarcomere. So the complete sarcomere is composed by this myosin and actin, means these are the contractile proteins. So firstly, how the actin molecules are involved in the contraction, how the myosin molecules are involved in the contraction, we are discussing individually, right. Firstly we are explaining about the actin. So this actin and myosin, these both are the polypeptide protein molecules especially. So these protein molecules are firstly actin. So these actin molecules having mainly two types of molecules, especially these are the troponin, and the second one is the tropomyosin, right. Firstly troponin, this troponin is again three types, three parts. So these are the troponin I, troponin T, troponin C. Troponin I means it is a troponin inhibitory molecule. Troponin T, it is a troponin tropomyosin binding molecule, and troponin C, troponin calcium binding molecule. So these three molecules are including in the troponin. And tropomyosin, this tropomyosin, it is a one long band like structure. This band like structure is present on the around the, that is filamentous molecules especially, right. Firstly, so it is the filamentous, means these are completely globe-like structures. It is the actin structure. This is actin filamentous. So these are the filamentous. F, filamentous means we are showing a symbol, that is F, right. Tropomyosin, it is completely present on the, that is along with on these filamentous molecules. So it is called tropomyosin. And these troponin molecules are present every interval of these actin molecules. So every interval means it is like this, completely along with this longitudinally these myofibrils especially. So these three molecules are, one is the troponin I, and the second one is the troponin C, and third one is the troponin T. So always the troponin T is bind to this tropomyosin molecule. Always this troponin C bind to the calcium molecules which are present in sarcoplasm. Always this troponin I inhibiting to the, that is muscle contraction. So these are the troponin tropomyosin structure, and it is the actin structure, right. So the function of the troponin C, it has a main role in the muscle contraction, especially when the nerve impulse are sent into the sarcoplasmic reticulum or sarcolemma. So immediately the sarcoplasmic reticulum is stimulated by the nerve impulse. So the sarcoplasmic reticulum has a cisternase. These cisternae are having a number of calcium molecules. That's why the cisternae are also called calcium storehouses. When the nerve impulse are land on the stimulate to the sarcoplasmic reticulum. Immediately cisterns are released to the calcium ions into the sarcoplasm. So always these troponin C molecules and always these actin molecules are always embedded in the sarcoplasm, right? So these troponin C molecules are bind to the calcium molecules which are present in the sarcoplasm. So immediately this troponin C is binding to the calcium molecules, this active myosin binding site will open. So it is the main thing. So if this troponin C cannot receiving to the calcium ions, right, that active myosin binding site does not open. So slightly like this, it is the actin. When the calcium molecules are binding to this calcium troponin C, right, after receiving the calcium ions, immediately it is open its binding site like this, slightly opened. So after that these myosin heads are just tilt towards these actin molecules. So it is the main process of this actin. So it is like this completely. It is completely present around the that is from proximal part to distal part of the muscle fiber. So it is the main structure of actin. Then we are going to the another contractile protein is myosin. So this myosin is also called dark band especially, and it is also called anisotropic band, means it cannot allow any light from it, right? So the complete myosin band has a number of approximately 200 to 300 myosin heads are present in each A band. So these myosin heads are the main function of these myosin heads are present. The main function of these myosin heads are attached to the actomyosin binding site especially. So for example the structure, it is the myosin band. So each myosin band has a number of approximately 200 to 300 myosin heads are present on this A band, right? So these myosin heads have a mainly two types of protein molecules. These protein molecules are heavy meromyosin protein molecules and light meromyosin protein molecules. So heavy meromyosin protein molecules are present on head region and also neck. These both are composed by heavy meromyosin protein molecules, and it is a tail from here to which are attaching with this band. It is the tail. So it is completely made by the light meromyosin protein molecules. It is the myosin structure. So these are the mainly called contractile protein. Both are involved in another one is that is 13. It is from immune system. So that is write a short notes on immunoglobulins. Nothing but antibodies. The main function of the antibodies is these are immobilizing, these are recognizing to the antigens, means foreign bodies, and these are recognizing to the foreign bodies and which are immobilizing to the particular foreign bodies. So the main function of these antibodies, that's why these are also called internal scavenger like phagocytes also. These antibodies are mainly produced by the mature B lymphocyte cells. So these when these mature B lymphocyte cells are converted into the functional B lymphocytes, these functional B lymphocytes are forms to the mainly two types of cells. These are the mainly memory cells and circulatory cells. So these always circulatory antibodies and memory antibodies are recognizing to the foreign bodies. So basically these antibodies are mainly 5 types. 5 types of antibodies are present in the organisms. These are the immunoglobulin A, immunoglobulin D, immunoglobulin E, immunoglobulin G, immunoglobulin M. These are the mainly 5 types of antibodies. Okay, right. So immunoglobulin A, immunoglobulin D, E, G, and immunoglobulin M. Immunoglobulin A, it is a dimeric antibodies. Dimeric antibodies. So dimeric means it is two antibody structure, especially it is like this. It is a dimeric. And D, E, G, these are the monomeric antibodies. Monomeric means which are having only one antibody, like this, monomeric. And immunoglobulin M, it is a pentameric antibodies. Pentameric means which is having 5 antibodies, especially like this. It is the immunoglobulin M pentameric antibodies, right? So immunoglobulin A, it is present in colostrum. It is a mother initial breast feeding of infants. And immunoglobulin D, immunoglobulin E, these are involved in the allergic reactions. Immunoglobulin G and immunoglobulin M. Immunoglobulin M mainly present in the formation of multiple alleles, means A, B, O blood groups. Immunoglobulin G, it is involved in the formation of anti-D antibodies. So it is the main function of these antibodies, means immunoglobulins. Firstly, we are discussing about the structure of antibodies, right? So the structure of antibody, it is commonly called H2L2 structure. It is the antibody structure, that is the H2L2 structure. H2L2 means H and L. H means it is the heavy chain, L means it is the light chain. Totally 4 polypeptide chains are formed to the single antibodies. So that is diagrammatically rough diagram, that is it is the antibody. These polypeptide chains are internally connected by the disulfide bonds, especially disulfide bonds. So this particular this space is a paratope. The main function of this paratope region is which is attaching to the, that is epitope. So epitope means that is the antigen structure. From this region is called FAB, means that is fragment antigen binding site. So which is called FC, means fragment cell body. So it is attaching to the, that is surface of the cell. And these are the disulfide bonds. These are the disulfide bonds. So here the L2 structure means these two long structures. It is the H2. So these are the totally 4 polypeptide chains. 1, 2, 3, 4. Right. From this region is called the heavy chain. This is L shape, and these are the H, means heavy. So these are also H2. That's why this antibody structure is called H2L2 structure. So it is the immunoglobulins and antibodies. It is the antibody structure. These are the types of antibodies. It is for the immunoglobulins and its function in the immune system.